The overall goal of this procedure is to demonstrate the ease of performing a Western blot when using appropriate products. Following sample preparation, proteins are separated based on size and then transferred to a solid phase. The proteins are then probed with antibodies and detected. The use of proper protocols and high-quality, cost-effective reagents produce enhanced Western blotting results.
Hi, I'm Jesse Lujan, R&D scientist at EMD Chemicals in San Diego, California. EMD is an affiliate of Merck KGaA in Darmstadt, Germany. Today we're going to show you a Western blotting procedure. So let's get started.
The first step in Western blotting is sample preparation. Begin sample preparation by pelleting the cells using low-speed centrifugation, such as 5 minutes at 2,500 times gravity. Following centrifugation, drain the cell pellets well. Resuspend the cells in cytobuster protein extraction reagent using 150 microliters per 10 to the 6 cells. Incubate the sample at room temperature for 5 minutes. Next, spin the sample for 5 minutes at 16,000 times gravity at 4 degrees Celsius. Following centrifugation, transfer the cleared supernatant to a fresh tube and proceed with analysis. After the protein concentration of the sample is assessed, prepare the sample for loading the gel. To denate